Okay, well, thank you. I'm Bill Wimley, faculty here in the Tulane School of Medicine. So my lab is interested in synthetic molecular evolution. We use synthetic molecular evolution to find peptides that do interesting and useful things. So what I'm going to talk about today are antimicrobial peptides. So we have evolved a family of peptides that basically a novel chemotype to fight against drug-resistant biofilm forming bacteria. Of course, we all know bacterial pathogens and acquired drug resistance is a problem. It's an exponentially growing problem, and some authors have stated that they can see the end of the antibiotic era. That's a bit of hyperbole, but it's a growing and severe problem in healthcare, and it's expensive, and there's a lot of money being spent. So we have used synthetic molecular evolution, that is iterative library design and screening, to find peptides that are we think host compatible, clinically relevant. This is a different approach than other people have taken in this field. So antimicrobial peptides are sort of a novel chemotype, right? They don't have a particular biochemical target in a bacterial cell. They don't target the ribosome or a synthetic enzyme like most conventional antibiotics. They target the entire bacterial membrane. And in doing this, they are not susceptible to, say, mechanisms of resistance that give bacterial pathogens, say, pan-drug resistance, like drug efflux pumps are ultimately can grow to become effective against any small molecule antibiotic. Peptides attack the cell membrane itself, permeabilize the membrane, breach the compartmentalization needed to keep the cell alive, and the cells die. So this is a very different sort of mechanism of action, and it's a useful chemotype to avoid some of the problems we're seeing with conventional antibiotics. Again, I don't have a lot of time here. I'm happy to discuss this in detail with anyone who wants, but the current family of generation of peptides that we've evolved has very good activity in vivo against all of these escape pathogens. These are some of the most common multi-drug resistant human pathogens. Not only do they have this sort of activity against these pathogens in vitro, but these activities are maintained in a very complex protein and cell-rich environment. So we actually screen for antimicrobial activity in the presence of highly concentrated human red blood cells as a surrogate for tissue and host cells. Almost all known antimicrobial peptides completely lose activity under these conditions. We've selected peptides that maintain extremely good antibiotic activity against a broad spectrum of gram-negative and gram-positive pathogens under these conditions. Now, a new antibiotic is useless, even a new chemotype is useless if organisms develop resistance to it quickly. So we took Pseudomonas aeruginosa, one of the trickiest and most difficult organisms, because it can rapidly evolve resistance to many different chemotypes, including some antimicrobial peptides. So we took Pseudomonas aeruginosa and we passaged it against 4 conventional antibiotics. And you can see, and these 10 passages include overnight passages, so many generations. And you can see we grow resistance even on a log scale. We grow resistance to these conventional antibiotics from day one. And eventually Pseudomonas becomes completely resistant to any of these conventional antibiotics. Against our peptides, we see no increase in resistance over many, many generations, passaged in the presence of sublethal concentrations of peptide. So we have some mechanistic ideas and an NIH grant to study the mechanism of this, what we call resistance avoidance, but it's a very important property in these peptides. We don't just have to kill bacteria sitting in a planktonic culture, but we have to be able to kill bacterial biofilms. So we've done some very nice work with biofilms. These are conventional antibiotics against Pseudomonas biofilms. Biofilms, as you know, are inherently resistant to many antibiotics just because of the physical restraint on the access of drugs to the cells, and because the cells tend to, biofilm tend to undergo a quiescent stage where they're not highly biochemically active. Against Pseudomonas biofilms. These conventional antibiotics have little activity. This is log reduction in viable biofilm bacteria. Again, against our peptides, the synthetically evolved peptides, we get very good activity against biofilms. Not only do we remove the physical biofilm by treating with these peptides, but we kill, up to 99.99% of the living bacteria in the biofilm. So we have very good biofilm activity. Now we've been working with Lisa Morisi, who you heard from earlier, and we've used this very nice wound model in mice where we take a deep puncture wound on the back of a mouse, and we infect it with drug-resistant bacteria, either Pseudomonas or MRSA, methicillin-resistant Staph aureus. These are organisms which have been engineered to be luminescent, so we can follow the bacterial burden in a single animal using whole animal imaging. Vehicle control. These are healthy adult mice, so they get basically a raging purulent pussy infection over 3 or 4 days, and then innate immunity kicks in, and they begin to clear it. So here's the course of an infection in a normal adult mouse. This is Pseudomonas. You can see for at least day 1, day 2, day 3, we have very high cell counts, very high luminescence from the luminescent bacteria. It eventually they begin to clear. You can see the plots of this luminescence as a function of time for Pseudomonas and MRSA. When these wounds are infected first and then treated with our peptides, we essentially knock back the bacteria by 3, 4 or more logs of bacterial burden, and at least on a linear scale, there's no evidence of bacteria there, being treated several times a day, 3 times per day over the first 5 days of the infection. So they're very good at treating a purulent infection in a wound. And this is a very, as I said earlier, this is a very cell-rich, protein-rich environment where a lot of peptide antibiotics don't work very well. In this experiment, we can also assay for biofilm formation. Of course, having bacteria sitting in this wound for 5 days, we get massive amounts of biofilms forming. And Lisa's lab has developed a very nice method of assessing biofilm formation, and treatment with peptides. I don't have the data here, but treatment with peptides completely eliminates detectable biofilm formation in the wound. Whereas vehicle control, the biofilms are everywhere. Okay, so current data package, we have a lot of preclinical data. We have a continuing evolution going on. The latest generation of peptides are quite good, the ones I just described. So we have a lot of in vitro efficacy data against a lot of microbes. We have a lot of data against clinical isolates of some of these worst pathogens, such as Klebsiella pneumonia and Pseudomonas aeruginosa and MRSA. So the latest generation of peptides has very good activity against a broad spectrum of clinical isolates. We have this animal model, and Lisa's lab is developing other animal models, which we will soon be using. We have activity against drug-resistant bacteria, against biofilm-forming bacteria, et cetera. Safety studies, we don't see much in terms of cytotoxicity. We can put a very large concentration of these peptides in an open wound in mice, and we don't see any adverse effect. We don't see any necrosis or evidence of damage to the wound. So it's well tolerated in this wound. Formulation studies right now, we've evolved these peptides to be highly soluble, so they work very well in saline solution. We're working on viscous time-release formulations that should be easy to develop for these peptides based on the physical properties. Peptides themselves are extremely stable. They're not like a typical biological drug like an antibody. So they're stable for years in lyophilized form and stable for up to 1 year in saline solution. Again, we're working on formulation. Manufacturing, well, we haven't gotten there yet, but I'm happy to talk about it. And yeah, that's me. And whoever wrote this it's kind of clever. We bring ideas to lives. So, thank you. And nothing has beeped at me yet. So
quick questions. We got 30 or so seconds. Yep.
So any evidence of allergic responses or anything like that?
We don't see any evidence of that in the wounds, and based on we haven't done the test of immune reaction to these peptides, but based on the literature, peptides of this size, 12 to 15 residues generally do not elicit an immune response. They're too short to be properly processed and displayed. Generally, these kind of peptides don't elicit an immune response, and they can be used over and over again. Has not been tested for this particular family, but we expect the results to be positive.